We take a deep dive into the mind of Cheri through laughter, logic, love, and clarity. Two voices meet where truth gets ready. The unMASKing Cheri Show.
Welcome back to the deep dive. Today we are opening a set of files that is maybe the most personal and specific we've ever tackled on the show.
It's pretty incredible stuff.
Yeah. You came to us with this curiosity, looking for a way to ground a self-identified neurodivergent identity in, hard biological evidence, and that is exactly what our source material delivers.
Precisely. This deep dive is a direct, really granular analysis of genetic source materials. We're focusing specifically on the DNA profiles of one individual, Cheri Harris, and her biological parents, Gene and Audrey.
Right. And we're not just looking at their whole genome.
No, that would be overwhelming. We have a very specific set of verified single nucleotide polymorphisms, or SNPs as they're called, and these are the ones that are strongly linked to various traits of neurodivergence.
So we're talking about things like autism spectrum traits.
Yes, and social emotional sensitivity, and also, this is crucial markers associated with giftedness. It's the whole package.
This isn't just theory then. It's not a broad population study. This is custom-tailored genomic insight. Our mission here seems really clear.
It is.
We are going to extract actionable knowledge about the inheritance patterns of these 5 specific genetic variants across these 3 family members. And the goal, which the source itself states, is to provide, and I'm quoting here, biological validation for Cheri's neurodivergent identity.
By tracing the genomic lineage. It's like genetic genealogy, but for neurological traits.
Okay, let's unpack this. We're moving beyond, behavioral checklists and subjective reports, and we're looking directly at the underlying neurological code.
And the data we have is incredibly specific. It's been pre-filtered. The whole foundation of this analysis is compiled from a source called the neurodivergent genetic profile.
So it's targeted.
Very. It's not a wide open search across millions of base pairs. It's a focused look at 5 scientifically recognized reference IDs, RSIDs, that were chosen for a reason.
And what's the reason?
2 reasons, really. First, because they are strongly associated with ND traits in major studies. And second, and this is the critical part, they were confirmed to be present in Cheri's own DNA.
Ah, okay. So that distinction is huge. These aren't just 5 random markers. They've been scientifically flagged as relevant, and then they were verified in this specific person's profile.
Exactly. It's not theoretical anymore. This is real genomic data, and it's showing us how specific traits literally line up across generations.
So it takes the conversation from this very general question of, is this kind of thing inherited, to a much more specific question.
Right, to how is this unique combination of traits inherited and expressed in this family line?
And that combination covers a lot of ground, doesn't it?
A huge range of neurological function. We're talking about everything from the basic structural development of the brain and its neurovascular support system, all the way through to emotional regulation and really high cognitive ability.
It's fascinating. The source document isn't just raw data then. It's not just a list of letters.
Not at all. What's so useful here is that it serves as both a foundational genetic record and an interpretive summary. It doesn't just give us the data, the A's, T's, C's, and G's. It gives us the context for why those letters matter in a neurodivergent profile.
That framing is everything. So before we jump into the specific markers, what does the document say about the sort of big picture genetic principles we need to keep in our minds through this whole deep dive?
It really emphasizes 2 core concepts that anchor this whole analysis. First, and this won't be a surprise to many, is heritability.
Right.
The source confirms what, extensive scientific literature has been saying for years. Autism and related neurodevelopmental traits are highly heritable. We are looking at a system that is literally designed to find these familial patterns, and as we'll see, they play out very clearly.
Okay, so that's the first one. High heritability. What's the second?
The second is the concept that truly defines the complexity of this whole thing, polygenic inheritance.
Polygenic, meaning from many genes.
Exactly. This is what makes a neurodivergent profile so much more complex than, say, a simple single gene disorder like cystic fibrosis.
This is the crucial point, right?
Absolutely. We have to stress this. These traits, they do not stem from a single gene mutation. There's no one autism gene or ADHD gene.
It's not that simple.
Not even close. They are the result of complex, cumulative combinations. You have many different genes, and each one contributes a small, subtle effect, and they all stack up.
So when we trace these 5 markers, what we're really looking at is direct evidence of a kind of familial ND wiring.
That's a perfect way to put it. It's a familial ND wiring that is consistent in different ways across both parents, Gene and Audrey, and then it manifests in Cheri. It's like a genetic mosaic. The pieces interact with each other, they don't just add up, and they create this very distinct neurological profile.
Which is why looking at the whole family, the trio, is so vital.
Yeah.
It shows you the full toolkit of variations that were available for Cheri to inherit. That sets the stage perfectly. So before we get into the actual genotypes, the letter combinations, we should probably make sure everyone's on the same page with the language of genomics we'll be using.
Let's lay that groundwork, but let's focus on why it matters to you, the listener. When we talk about genetics at this level, we're dealing with these things called single nucleotide polymorphisms, SNPs, it's pronounced SNPs.
SNPs.
An RSID, which you'll see listed for each marker, is basically just a catalog number. It's a stable, known location in the human genome where a single letter of the DNA code, A, T, C, or G, commonly varies from person to person. It's just a specific variation point.
And these 5 RSIDs, these 5 locations, they aren't random.
Not at all. The source flagged these specific locations because they are highly relevant to ND traits based on rigorous studies, these massive genome-wide association studies, or GWAS data.
And these variations, these different letters, they don't necessarily cause neurodivergence, right?
That's a key distinction. They don't cause it, but they absolutely influence the likelihood of it. They influence the expression, the intensity of certain traits. Things like how resilient are your synapses, or how efficiently does your brain use its energy.
That's all about tendencies and predisposition.
Yes. The source's methodology is targeted. It's only looking at these 5 locations. And that selectivity is what makes the interpretation so powerful, because we're focusing only on the variants that the scientific community has repeatedly linked to the exact profile we're talking about.
Okay, and for each person, their specific code at that location, their genotype, is shown by 2 letters, one from each parent.
Right, so you'll see a pair of letters like AA or maybe TC. If the letters are the same, we call it homozygous. If they're different, it's heterozygous. And that tiny detail, homozygous versus heterozygous, it tells an immediate story about inheritance.
And we're going to see how the homozygous profiles provide a kind of stability, and the heterozygous ones introduce nuance.
That's the perfect way to think about it. So let's start with that stability. Let's look first at the pattern of strong inheritance. These are the markers where the genetic profile is just remarkably consistent across all 3 family members.
This suggests a really stable, deeply inherited foundation for some of these core neurodevelopmental traits.
It does. First up, we have SNP1, RS167771. This one is associated with the RELN gene.
RELN. Okay, what does that one do?
The RELN gene, or RELN, it's foundational. It plays a really critical role in brain development, especially in how neurons migrate during fetal development. It makes sure the brain's architecture gets assembled correctly.
So it's like the master architect for the brain.
In a way, yes. And even after development, RELN is vital for synaptic plasticity. That's the ability of your brain's synapses to get stronger or weaker over time. It's the biological mechanism for learning and memory.
And changes in this gene are often studied in relation to autism, right?
Yes, both autism traits and schizophrenia, because it really governs the fundamental structure of how the brain is connected.
Okay, so the family profiles here, what do they look like?
They are definitive. Cheri is AA. Gene, the father, is AA. And Audrey, the mother, is also AA.
Wow, so it's completely consistent across the board.
A textbook example of fully inherited homozygous consistency. All 3 of them have the exact same variant at this really important structural node. So when the source interprets this AA genotype, it links it directly to a consistent neurodevelopmental profile linked to autism.
Okay, but what does consistent neurodevelopmental profile actually mean in the real world? For a neurodivergent person, especially with a strong, consistent homozygous profile like this, how does that AA manifest day to day? It's not just about brain architecture, is it?
That's the key question. It really speaks to the fundamental nature of how they process information.
Yeah.
This specific AA variant can often be associated with differences in things like neural pruning and learning flexibility.
Okay.
So if the RELN pathway is consistently configured this way, it suggests the brain might favor highly specific, really robust pathways, but pathways that are perhaps less flexible.
So like a superhighway instead of a network of back roads.
That's a great analogy. This wiring could contribute to strong systemizing abilities, incredible pattern recognition, which is a characteristic strength in many ND profiles. But the flip side is it could also contribute to rigidity or difficulty adapting quickly to new things because the foundational architecture is just wired for deep stability.
It's the foundational architectural style. It's like if everyone in the family built their house on the same type of reinforced concrete foundation, you know that the house, no matter what color you paint it, is going to have a certain structural integrity, a certain resistance to major shifts.
Exactly. This AA profile from the RELN gene establishes that stable foundational ND wiring for Cheri.
Okay. And the next marker gets even more specific.
It does. It drills down even further into trait specificity. We move from the overall structure to attention. SNP2 is RS4307059, and it's located near a gene called MSNP1AS.
And this one's association is explicitly tied to strong autism susceptibility. The source says it particularly affects language, attention, and communication processing.
Yes. And once again, we see absolute homozygous consistency across the entire lineage. Cheri is TT, Gene is TT, Audrey is TT.
Fully inherited, double-dose TT from both sides. This is where the source's interpretation delivers a really major insight, isn't it?
It's huge. The source directly interprets this TT genotype as a strong autism association, specifically tied to monotropic focus and communication traits. This is where the genetic data directly validates a core behavioral trait.
That is a profound linkage. Monotropic focus, that tendency to focus attention so deeply, often just on one or two things, is a cornerstone trait for so many autistic people.
It is. So when the source links the inherited TT genotype directly to this, it gives it massive biological weight.
But how certain can the source be that this specific TT genotype at this specific RSID really means monotropic focus?
Well, the strength of that claim comes from the aggregated GWS data that inform this profile. Now, of course, no single gene causes a behavior, but this particular SNP has shown up again and again in studies that look at intense focus and the communication styles that go along with it. Functionally, this TT configuration suggests a neurology that's optimized for depth over breadth when it comes to attention.
So the brain is dedicating immense processing power and energy to one channel of information.
Exactly. Which is a powerful, powerful advantage for developing deep expertise and mastery in a subject.
But, like with all ND traits, there's another side to it. If the brain is wired for such deep dedication to one focus, what are the inherent challenges that come with that specific wiring?
The challenge lies in transition and regulation. If all your neurological resources are heavily invested down one track, shifting your focus becomes biologically costly. It can even be painful.
And that shows up as what?
It manifests as that common neurodivergent difficulty with task switching, difficulty engaging in small talk that's not related to your focus, and potential rigidity when your routines are interrupted.
So the TT genotype provides a kind of biological validation for the high energy cost of switching contexts.
It really does.
Yeah.
Which is a major factor in managing life with an ND profile. It's not a lack of willingness to switch tasks. It's a very real, very high neurological hurdle to overcome.
It essentially confirms that Cheri is genetically wired for a specialized processing style. And the fact that both of her parents carry the exact same homozygous profile that suggests this specific variant has been stable and, evolutionarily successful in this family for a long time.
That's a very good point. Now let's move to the third consistent marker. This one deals with how the brain actually powers these intensive processes. We're talking about SNP3, RS3025039, which is related to the VDGFA gene.
The association here is less about behavior and more about the physical support systems in the brain, right? Neurodevelopment and, crucially, brain blood flow neurovascular support.
Yes, VGFA stands for vascular endothelial growth factor A. It's a powerhouse molecule. It's essential for creating new blood vessels and maintaining the ones you have, especially the complex capillary network that supply the brain with oxygen and glucose.
And that's critical for high cognitive function.
Absolutely. Robust neurovascular support is critical for maintaining high, sustained cognitive function, especially during periods of high cognitive load, intense concentration, or stress.
Okay, so let's look at the genotypes here, because this is where we see the first little difference in the mother, Audrey. But the resulting profile in Cheri is still strong.
Right. So Cheri is CC, Gene, the father, is CC, but Audrey, the mother, is TC.
So she's heterozygous.
Exactly. This is a perfect illustration of how inheritance works. Audrey, being TC, carried both the C allele and the T allele. Gene, being homozygous CC, could only pass on a C.
So Cheri got the C from Gene.
And statistically, she also inherited the C allele from Audrey, which resulted in her having a homozygous CC profile.
So even though her mother carried that T variant, Cheri still ended up with the double dose of C and the interpretation from the source.
The source ties the CC profile in Cheri to strong neurovascular support in ND profiles.
Okay, let's go deep on the functional impact here. How does robust brain blood flow actually manifest in a person who is already wired for monotropic focus and high cognitive intensity?
This CC profile provides a biological engine for that ND brain. Think about it. If your brain is naturally wired for that intense TT focus, it requires a phenomenal amount of energy. That energy is supplied via oxygen and glucose delivered by blood flow.
So the CC variant is like having a better fuel pump.
That's a great way to put it. It suggests optimized neurovascular capabilities that can help sustain those intense processing states, that hyperfocus for longer periods without an immediate crash.
So we could speculate that this CC profile might allow Cheri to push through mental barriers that other people might hit much more quickly. Does it translate into better endurance against mental fatigue, or maybe faster recovery from cognitive drain?
Precisely. If we link this CC result to the TT monotropic focus, we see a totally integrated system. The focus dictates the mode of processing, and the vascular support dictates the stamina for that processing.
Oh, that makes sense.
It suggests that while the individual might still experience burnout from external stressors or task switching, their internal capacity for intellectual endurance during highly focused work is robustly supported at a physiological level. It's almost like a genetic marker for intellectual stamina.
So to just summarize this whole section, we've basically established a fundamental structural genetic foundation for Cheri's profile. It's based on 3 markers that are either completely consistent,
The AA for foundational wiring and the TT for monotropic focus.
or they converge into a strong homozygous profile, like the CC for neurovascular support.
Yes. This triplet of SNPs confirms the familial ND wiring at a structural, an attentional, and a supportive level. These are the stability points of her profile.
Right. And now we move into the markers that show more of a mix.
Indeed. Now we get to the markers that demonstrate mixed inheritance. These next 2 SNPs are where we see the introduction of heterozygosity. They show how different alleles from the parents can combine, and they really reinforce that essential concept of polygenic inheritance.
It's not just one inherited trait. It's the layering of different variations that creates the unique and often, paradoxical individual profile.
Exactly. And we're going to transition from that hardwired focus to the softer skills that often define so much of the ND experience, social and emotional awareness.
So we're at SNP4, RS53576. This is the famous one, right? Associated with the OXTR gene.
This is arguably one of the most widely studied SNPs in all of social neuroscience. The OXTR gene codes for the oxytocin receptor.
Oxytocin, the love hormone, or a cuddle chemical.
That's what they call it. It's a neuromodulator that's crucial for social bonding, trust, empathy, and also for managing our stress responses.
Yeah.
So variations in this gene are studied extensively because they significantly influence social cognition and stress reactivity.
Both of which are central aspects of the neurodivergent experience.
Absolutely.
So this is where we see a distinct heterozygous profile in Cheri, which is a direct result of inheriting different alleles from her parents.
That's right. Gene, the father, is GG. Audrey, the mother, is AG. And that means Cheri is AG.
So she inherited the G allele from Gene. It was the only option he could pass on. And she inherited the A allele from Audrey.
And that AG combination is the absolute key to the interpretation here. The GG profile, like her father's, is typically associated with lower social stress, maybe greater resilience. The A allele, on the other hand, is often linked to heightened emotional awareness.
And the source specifically links this AG genotype in Cheri to, what was it?
Emotional sensitivity and higher empathistris.
Higher empathy and higher stress. We're talking profound empathy here. How can one tiny variation, just the presence of that single A allele, dictate something so complex and dual-edged?
Well, it's thought to relate to the density or the efficiency of the oxytocin receptors in the brain. The A allele often results in fewer or less efficient receptors.
Which sounds counterintuitive. Fewer receptors means more empathy.
Paradoxically, yes. It can lead to 2 outcomes. First, a very high need for social connection and bonding. And second, A state of hypervigilance or hyperattunement to social cues may be driven by an unconscious attempt to compensate for that receptor difference.
So you're constantly scanning the room, trying to pick up on everything.
Exactly. And that high attunement is experienced as profound empathy. You're literally absorbing other people's emotional states. But because the system is running at this heightened, maybe compensatory level, it dramatically increases your own stress and vulnerability to sensory or social burnout.
Wow. If we connect this to the bigger picture, this particular inheritance pattern could explain the biological root for that intense stress so many highly sensitive neurodivergent people report after social interaction.
It provides a very strong biological basis for it, yes.
Does the source offer any insight, even theoretically, into how you might mitigate that high stress response knowing its biological root?
Well, the document doesn't explicitly prescribe treatments or anything like that, but the implication for you, the listener, is clear. Biological validation informs strategy.
Right.
If you know the AG profile predisposes you to high stress and empathy, then your strategy has to focus on proactively managing your social environment. That means ensuring you get sufficient alone time, setting clear social boundaries, and developing robust emotional regulation tools.
It makes sense that someone with this profile would really benefit from environments that minimize social ambiguity and maximize predictability.
Absolutely. It validates the need for retreat and recovery. This marker, unlike the first three, is all about the interaction of two different alleles, and it leads to this really complex trait, high empathy and high stress reactivity. It's a defining characteristic of Cheri's ND identity.
And that leads us perfectly to the fifth and final SNP, which balances the narrative beautifully by focusing on cognitive strengths.
Yes. SNP5 is RS7793837, which the source explicitly links to giftedness.
The associated traits here are fluid intelligence and working memory. So the ability to think abstractly, solve new problems without prior knowledge, and hold and manipulate multiple pieces of information in your head at once.
These are traits often found at the upper end of the cognitive spectrum, and they frequently co-occur with neurodivergence.
So looking at the genotypes, Gene the father is AA, Audrey the mother is AT, and Cheri is AT.
So Cheri inherited the A allele from her father and the T allele from her mother. And the source notes that the T allele is specifically associated with high cognitive ability in large-scale genome-wide association studies.
This is hugely significant.
It is. It immediately reinforces the definition of neurodivergence as a spectrum that inherently includes profound, high-functioning intellectual variance. The T allele she inherited from her mother is a key component of this specific familial profile. It suggests that the ND wiring in this lineage is intertwined with exceptional cognitive capacity.
Okay, so what are the functional implications of having this AT profile for fluid intelligence coexisting with that TT monotropic focus profile we talked about earlier?
Yeah.
How do those two work together?
It creates a kind of specialized processing machine. The fluid intelligence from the AT profile provides the engine for complex, abstract problem solving. Meanwhile, the monotropic focus from the TT profile provides the dedicated, sustained pathway for applying that power.
So one provides the horsepower, and the other provides the laser beam to direct it.
That's a perfect analogy. This combination suggests a brain that's optimized not just for solving problems, but for intensely specializing in complex, highly specific domains. And that is often the foundation of professional or academic success for many neurodivergent adults.
It confirms that this familial ND wiring isn't just about potential challenges. It fundamentally includes genetic indicators of unique, strong cognitive abilities. The T allele inherited from Audrey ensures that the biological validation of Cheri's identity incorporates both that high stress sensitivity and this exceptional mental agility.
And this complexity just underscores the polygenic reality we talked about at the beginning. We are analyzing a profile that combines structural differences from SNP1, a specific processing style from SNP2, the stamina to fuel it from SNP3, social regulatory challenges from SNP4, and now intellectual firepower from SNP5.
We have mapped out 5 distinct markers, 3 that define developmental consistency and foundational autism traits, 1 defining social-emotional sensitivity through mixed inheritance, and 1 defining high cognitive strength. So let's move into the synthesis. What does this 5-marker composite profile truly tell us about the Harris family lineage and Cheri's unique neurological identity?
Well, we can now definitively address the central mission of the source document, which was validating the genomic lineage. The document confirms that Cheri's neurodivergent traits have a clear, traceable, and really complex genetic origin that integrates the contributions from both of her parents.
This is the direct evidence of inheritance. It shows that the parents, Gene and Audrey, together carry the entire spectrum of these 5 specific neurodivergent variants, even if they express them differently themselves.
Exactly. The sources provide evidence that the familial ND wiring is consistent, as seen in both Gene and Audrey, even where their specific genotypes differ slightly.
Like Audrey being TC for VGFA and AT for giftedness, while Gene was CC and AA for those same markers.
Right. Cheri's profile is the sum of those possibilities, but it results in her own unique stacking effect. She gets the CC for neurovascular support combined with the AT profile for high intelligence, and critically, that double-dose TT for monotropic focus.
So Cheri's profile is a clear, identifiable blend of her parental markers, and it results in specific neurological variants that line up directly with her lived ND traits.
The TT genotype for intense focus, the AG for heightened emotional sensitivity, the AT for fluid intelligence, these are biologically confirmed influences on her neurology. It gives tremendous weight to her self-identification.
The strength of this deep dive really lies in moving away from any of those lingering single-gene myths. We should review the evidence for polygenic inheritance one more time, but this time by talking about how these 5 markers actually interact.
Let's do that. Let's break down that interaction.
Okay, so we looked at RS167771, the RELN gene, which governs broad brain development.
Then RS4307059, which influences that intense focus.
Then RS4360357A, regulating the brain's support systems, the stamina.
RS53506, the OXTR gene, which dictates social and stress sensitivity.
And finally, RS77933837, which drives that high cognitive ability. When you look at that whole 5-marker profile together, that is one intricate blueprint.
It is entirely integrated. Consider the interplay. The high cognitive ability from RS77933837, it needs the foundational stable processing style from RS167771 and the physiological stamina from RS302F25039 to even be deployed effectively.
Right, you need the engine and the fuel.
But then, when that powerful cognitive engine is channeled through the very specific intense processing method of monotropic focus, the TT genotype, and that same individual has to navigate the world with a heightened social and emotional filter from the AG genotype, the intensity of their whole experience is just multiplied.
So the very genes that confer exceptional intellectual power can simultaneously create a magnified vulnerability to the social environment. The capacity for deep processing clashes with the high cost of social engagement. This is the biological definition of the ND paradox.
That's it, exactly. If any one of those pieces were different, let's say Cheri had inherited the non-sensitive GG allele for OXDR from both parents, her manifestation might shift. It could be high cognitive ability with low social distress. This complexity is why polygenic traits are so difficult to model in general, but so illuminating when you can study them in the context of an established family line.
And this brings us right to the practical application for you, the listener. The source document itself instructs that this data should be cross-referenced with psychological, behavioral, or medical records for what it calls integrative insight.
This is the so what, and it's critical. Genomic data alone does not dictate destiny, but it provides a framework for self-understanding and for advocacy.
How so?
Knowing that there is a biological predisposition to intense focus, the TT, and high emotional sensitivity, the AG, it moves the discussion of burnout or social difficulty away from being a moral failing or a purely psychological issue. It becomes an issue of biological resource management.
It allows you to integrate that genetic confirmation with your lived experience. It provides that biological validation for an identity that is, let's be honest, often dismissed as being purely behavioral.
It becomes an internal compass. If you know you are CC for exceptional neurovascular support, you know you can probably sustain certain tasks for a long time, but you also know that your AG sensitivity means you absolutely have to schedule recovery time after deep social immersion.
It shifts the internal narrative from why am I like this to this is how I am wired, and this is why my needs for accommodation and specific structure are non-negotiable.
And that shift, grounded in hard science, is incredibly powerful for self-acceptance and for effective self-advocacy.
And what's more, the source provides a future-oriented instruction. It encourages the listener to think about the broader lineage. It says this data can be used to explore expanded inheritance patterns if additional family genomes are added later.
This raises a really important question. How might tracing these same 5 markers through siblings or aunts, uncles, cousins further refine our understanding of risk and resilience within this family lineage?
Because the spectrum is so wide, comparative data is gold.
It is. Imagine if Cheri had a sibling who also inherited the A allele for RS53576, so they have the emotional sensitivity, but maybe they inherited a different allele at the RELN marker, suggesting greater neural flexibility.
Their profile would be one of high social sensitivity, but maybe without the same structural rigidity we see in Cheri.
Or conversely, what if an extended family member had the CC neurovascular stamina marker, but they didn't carry the giftedness T allele? Their endurance might be applied to non-intellectual, highly consistent tasks.
So analyzing the full family tree would allow researchers, or the family themselves, to see which specific genetic combinations lead to which specific outcomes.
And that helps to separate the necessary foundational wiring from the more nuanced additive or even subtractive effects of the other SNPs.
It turns current profile from just a singular data point into a vital comparative baseline for understanding the genetic lottery of neurodivergence, refining our understanding of what the source calls risk and resilience genes.
The exact same TT variant that might contribute to difficulty transitioning in a traditional school environment could contribute to exceptional performance as a software engineer or a specialized historian. Tracing the inheritance across multiple people helps separate the core wiring from the environmental adaptation and performance.
Let's bring this deep dive to a close by reviewing the results of our analysis. We successfully dissected the profiles of Cheri, Gene, and Audrey to confirm the strong heritability and polygenic nature of neurodivergent traits in the Harris family line. We provided that biological validation for Cheri's identity.
We established a clear, consistent genetic lineage in 3 foundational markers. We confirmed traits associated with developmental stability, intense attention, and processing stamina. That's RS167771AA, RS4307059TT, and RS3025039CC.
And the identification of TT for monotropic focus is a particularly key takeaway for you listening, as it directly links an often misunderstood behavior to verifiable biology.
Then we explored the mixed inheritance markers, which added that crucial depth and nuance to Cheri's profile, particularly the AG genotype for emotional sensitivity and high stress from RS53576, which gives a biological basis to her social experience.
And we also confirmed the presence of the AT profile, where the T allele is tied to high cognitive ability and giftedness, from RS7793837. This ensures the validation includes a strong component of intellectual strength.
The analysis at the end of the day fundamentally underscores that the neurodivergent experience in this family line is the result of a complex, integrated genetic architecture inherited from both parents. That specific combination, high focus, high stamina, high intellect, high sensitivity, provides a robust explanation for Cheri's specific lived identity.
So if the DNA provides biological validation for a neurodivergent identity, what does that mean for how society defines and supports neurodivergence? We have to move past defining it solely by its deficits.
It suggests we have to move past generalized diagnoses and start supporting the specific neurological requirements dictated by these unique genetic profiles. If someone is genetically wired for high emotional sensitivity and monotropic focus, their support needs, whether in school or at work, must reflect that specific intensive combination.
They need environments that utilize the focus without constantly triggering the sensitivity. The ultimate final thought to leave you with, building on these sources. How does the presence of high cognitive ability markers, like that T allele in RS7793837, intersect with the traits linked to autism susceptibility and daily function?
That is the ultimate question.
Does the high fluid intelligence compensate for the social challenges? Or does the intensity of focus required to use that high intelligence actually exacerbate the social-emotional stress, driving the individual into deeper and deeper burnout cycles?
It's the ultimate question of adaptation, how the specific genetic hand you are dealt plays out in the specific environment you have to navigate. And knowing the code is the first step to mastering the environment.
That's a deep dive for another day. Thank you for joining us.
Always a pleasure to explore the code.